So it's my great pleasure and joy to welcome you all today to our Zika Plan dissemination event. Zika Plan stands for Zika Preparedness Latin American Network. Our consortium brings together 25 leading research and public health organizations from Latin America, from North America, Africa, Asia, and Europe. And we are united to fight Zika virus and to build long-term outbreak response capacity in Latin America. So funded by the European Commission, we developed 15 work packages to address the complex and diverse issues of Zika. And to mark the culmination now of almost 5 years of transnational efforts, we are now hosting a series of open access public webinars featuring some of the key findings of our consortium. So today is the first of such events. In fact, it's the first of 3 events. Although, like you all, we would have preferred to meet in Brazil, where we had originally planned to hold a 3-day conference. But alas, due to COVID, we had to be creative and we came up with 3 online webinars. Herewith, I would like to thank the Global Health Network for hosting these webinars. In particular, many thanks to Raman Preet from Umeå University and to Bonnie Baker from Oxford University, who have been pulling together the strings to make this webinar happen today. So now let me hand over to Bonnie for some short housekeeping announcements.
Thank you, Annelies, and welcome everyone. Just a few short housekeeping rules to make you aware of. This webinar is being recorded and the recording will be made available on the platform shortly after our session. The participant videos and microphones have been disabled. And please use the Q&A box to introduce yourself and post your questions throughout the session. There will be plenty of opportunity for your questions to be addressed, and we look forward to hearing from you. Thank you.
So thank you, Bonnie. So maybe back to the first slide. So this first seminar will have 3 topics. One is dedicated to congenital Zika syndrome, and the second topic is around the biology of Zika viruses, and the third is on the mosquitoes, the vector issues. But before we start with the science, let me hand over to our keynote invited speaker, Dr. Gustafu Mato. Gustafu is a social psychologist from the Oswaldo Cruz Foundation in Brazil, and he's coordinator of Zika Social Sciences Network since 2016. And he's also part of our sister or partner consortium, Zika Alliance. As Zika has affected communities and had a major social impact, it is only appropriate to kickstart our webinar with someone who understands the Zika-led social disruption, such as Gustafu Mato. Gustafu is also very much, has been involved in this whole process of the 3 consortia. And so thank you, Gustafu, for agreeing to give this talk. Over to you, Gustafu. Thank you.
Sorry. Thank you, Annelies. So thank you for Zika Plan as well. So it's an honor to be here and to have this opening session to highlight the social science contribution and also to explore some social and humanitarian issues related to Zika outbreak, especially in Brazil. So I will share my screen with you now. So the idea here is very briefly to give an idea of the repercussions on different dimensions and social dimensions, scientific dimensions, and also public health dimensions from Zika outbreak. So firstly, I would like to share some initial statements from social science. Zika represented and still is an uncertain issue in public health. As you know, there are many unanswered questions about Zika outbreak, his causality, repercussions, neurodevelopment, and also socio-economic impact, especially on women and children. Science, health, and society are intrinsically entangled in the Zika outbreak and its repercussion. This is the way that social science understands this kind of a phenomena. It's impossible to separate science, health, society, and many other dimensions in this problem. Social science are focusing on contexts, dynamics, historical, social, political, geographic, and cultural. I would like to say that we don't have one Zika, but many Zika around the world. Global, regional, and local inequities characterize the Zika epidemic and response. This is also important issue that I would like to develop a little bit with you. Zika as a political and social analysis of the invisibility of multi-disabled children and people in our world. So we have a very good colleague, Hannah Cooper from London School of Hygiene and Tropical Medicine. And she is a very good partner in our evaluation of social impact of Zika outbreak in Brazil. And she used to say that we have 1 billion person living in invisibility if we are thinking on disabled people. And after COVID-19, but getting back to Zika outbreak, it's important to say at the moment, at this very moment, every health emergency is sanitarian and humanitarian crisis. It's not possible to focus on just on vector and a virus and infectious disease as a biological phenomena. We have to focus on a complex phenomena involving society, political, diplomatic issues as we face it during COVID-19. So, also very briefly, I would like to share with you some topics that we are focused in our Zika Social Science Network, especially in the Zika Alliance Consortium that I coordinate with Jocelyn Hood, the work package focused on social science. Historical and social entanglements related to Zika emergency. Identify and analyze coordination and cooperation mechanism to contribute to a comprehensive health and social system. It is also important to say. Map the production and circulation of different discourses about Zika virus epidemic. That's why I would like to say that we have many Zika. Understand the dimension of Zika epidemic in context and on situations of vulnerability, analyzing through an intersectional perspective, gathering gender, race, and social vulnerabilities. Analyze the main epidemic repercussions of Zika in response of the health system and services in Brazil. It's also important to say that we would like to explore it in many countries in Latin America affected by Zika. Analyze and debate equity in scientific cooperation among national and international partners and the stakeholders related to Zika research in Brazil. There are many issues to explore in this situation. Improve digital methods in health using scientometric, autometric, and digital methods to map public understand perceptions and engagement on the Zika epidemic, and evaluate the economic impact of Zika outbreak on families. So this is the social science spectrum. So this is, would like to say that we can have a cross-cutting perspective on social science, since clinical and health care through bioethics and fieldwork research. So I would like to highlight some 3 points with you. One about uncertainties. So Zika shows that we have a kind of performatic of global health uncertainties, especially the whole of global health institutions around the world to respond to Zika outbreak. Public health uncertainties as well, especially in low and middle income countries, and clinical uncertainties. It's also involving to research and also assistance, because now in Brazil, the continuing of the health assistant depends on the continue of the research activities. So this is also important to say. So about scientific cooperation, this is the second issue that I would like to explore with you. So I have this paper published in early 2020 about Zika research response and cooperation. So we can say here that USA, as usual, have more than 50% of the publications, and the number of cooperations is related to Brazil. The Brazil is the second country that most publish on Zika during the 5 last years. So how we can continue like that, how we can find sustainability along time to keep this position, and what we could say about the other Latin American countries. In this image, we can see other Latin American countries in this most published countries through Zika. This is another work that I would like to share with you. It's from Paulo Paita and his team, also from Zika Alliance. You can see here the confirmed cases in Brazil and the concentrate of the cases in the northwest of the country. So since 2015 to 2017. So, and this is the distance and this distribution of health unities, how challenging is to attend all the families and children infected by Zika in a comprehensive way. So in a fast ways as well. So the distribution is very unequal in 3 selected states, in Rio Grande do Norte, Paraíba, and Bahia. So this is a big issue to face in an unequal country as Brazil. And also health professional availability. So we can see here a very good example of psychotherapists, but if you look at the important professional involved with Zika assistance, we don't have enough neurologists to take care of our children and how to evaluate the impact of the lack of assistance, especially in poor states in Brazil. So, and another issue that I would like to highlight with you is the gender, race, and social structure of inequalities. So it's not just about Zika, it's about a social structure in Brazil and many other countries. And we have many talking and analysis and analyzing this issue. The whole and overwhelming of women and caregivers, the sexual and reproductive rights, the economic impacts on family and women, the political harassment on women's rights that we are living at the moment in Brazil, where human rights are under attack in our federal government, especially the funding to science, technology, and healthcare. The invisibility on race, social, gender, intersectionality. the whole of women in Zika scientific leadership and scientific production, funding, and acknowledgement. So there's many topics that I would like to highlight and to share with you to think how to face and go ahead with this kind of inequalities. So some conclusions. Nowadays, in the aftermath, it's impossible to see that children affected by Zika virus have been framed and treated, to a large degree, as another segment of neglected population group, inscribing further inequities in Brazil and global health genealogy. COVID-19 difficulted health access and social protections among families. We are living a kind of drama during COVID pandemic. If you think that we are living another important and immense epidemic after 5 years of Zika outbreak. So this is the second impact for these families. When WHO declared the end of public health emergency of international concern, Zika funding was over. All of us knows about this. Health access in the most Latin American countries still depends on research funding to support cohort studies and healthcare. Danger issues in scientific production and social protections still are invisible for governments and funders. So this is some topics that I would like to share with you and probably could stimulate further discussions during this webinar. So this is my tribute to women in science and women that are resistant in this very moment in Brazil. You can see here in this picture many women that are leading scientific research groups in Brazil, and also the very brave women that fighting and represented mother associations in Pernambuco, Bahia, and Rio de Janeiro. And other colleagues here that most of them are watching this webinar. So thank you very much. If you would like to share, to see other production of our group, this is the link. It's based on the Global Health Network website, Zika Social Science Network. Thank you very much.
Thank you, Gustafu. You captured the pertinent issues brilliantly, and thank you again. And I also want to thank you that you will carry on with the legacy of Zika Plan and Zika Alliance and Zika Action, even after our projects end. And so that's also very important. Now, let's watch a film together. This film is entitled Hope After Zika and was produced by Zika Plan partners at a London school in 2018. The film is about community engagement and support and will set the scene why pioneering research for Zika was and still is so important. Thank you.
João e Verônica moram em Salvador, Bahia, com sua filha Liz, que nasceu em 2015.
Tive uma gravidez tranquila, não mostrou nada. Depois que ela nasceu, foi que os médicos vieram conversar. Quando a gente teve a certeza de que ela tinha microcefalia, ela já estava quase com 5 meses. Eu particularmente, me senti emotiva, assim. Eu imaginava, com a estimulação precoce, vai demorar, mas ela vai fazer tudo.
Liz, como milhares de outras crianças de sua idade, nasceu com a síndrome congênita do vírus Zika. Sua família é uma de muitas pelo Brasil que frequenta um grupo local de suporte onde pode compartilhar sua experiência e receber conselho. Os grupos são assistidos por um profissional de saúde e apoiados por pais voluntários como Silvia, cuja filha também foi afetada pela síndrome congênita do vírus Zika.
Quando eu soube do projeto, eu achei maravilhoso. E a família não, a família tem uma experiência e o projeto traz isso. Ele junta a experiência da família com a experiência do terapeuta. Os assuntos discutidos no grupo são vários. A gente tem uma sessão de posições e movimentos. Temos a sessão de comer e beber, que é a que eu mais gosto, mais me identifico.
Depois que a gente começou a frequentar o grupo, mudou a nossa forma de alimentar, porque ela se joga muito para trás e às vezes ela sentia dificuldade em engolir e a gente deixava, mas a gente aprendeu no grupo que isso dificulta muito, né? Ao beber uma água, a engolir a comida.
Os grupos também oferecem uma fonte importante de suporte psicológico para as famílias. Depois do choque de um diagnóstico, há um período de ajuste para os pais que pode ser bem difícil.
Nós contamos como descobrimos que as nossas crianças são portadoras de Zika, como nos sentimos naquele momento em receber uma criança portadora do Zika vírus. Falamos sobre a nossa aceitação, procuramos entender se, por exemplo, eu ainda estiver nesse momento de luto, né, as facilitadoras nos orientam a entender que essa mãe precisa de mais tempo ou que essa família precisa de mais tempo para passar por esse momento de luto. Ou não, que ela já venceu o luto. Como foi que ela venceu? Quais foram as estratégias que essa família utilizou para vencer esse momento de luto?
Depois que a gente começou a frequentar o grupo, a gente houve um sentimento de apoio, houve um sentimento de que nós não estamos sozinhos.
Você não precisa ficar só. Você pode se conectar com outras famílias, compartilhar suas experiências, receber apoio ou oferecer apoio para outros. Comece entrando em contato com o Movimento Zika 21 2239 9379 ou visite o website www.movimentozica.org.br. Você pode se juntar a um grupo de suporte ou até começar um grupo na sua área. A vida para sua família ainda pode ser gratificante e cheia de amor se a sua criança é afetada pelo Zika.
As crianças nos inspiram a ser humildes, né? A refletir, fazer uma autoanálise sobre a nossa existência, o que de fato tem valor para a gente, o que de fato é importante. É tão bom, é tão lindo ver o sorriso de uma criança, é tão lindo sentir o toque e saber que a criança está bem e que ela é amada por todos. Eu acho que ela veio para confundir os egoístas e trazer humanidade para a gente.
Well, that was an impressive film and reminds us and motivates us to continue in our Zika research to make sure that we do better when it does return. We will now start with the science, and it's my pleasure to introduce Associate Professor Elizabeth Brickley to you. She is Associate Professor at the London School of Hygiene and Tropical Medicine and will be chairing the first session on the full spectrum of congenital Zika syndrome. Thank you, Elizabeth.
Thank you, Annalise. And thank you everyone for joining us. So today I'll be talking about Zika virus infection during pregnancy and congenital Zika syndrome. When the cluster of children born with microcephaly was detected in the northeast of Brazil in late 2015, a task force of Brazilian public health experts known as the MIRG, the Microcephaly Epidemic Research Group, was rapidly assembled to get to the cause of the outbreak. The MIRG researchers, including Professors Celina Turki, Ricardo Jimenez, Democrito Miranda Filho, Talia de Araujo, Laura Rodriguez, Ulysses Monterroyos, Viner de Souza, and Maria Albuquerque, among others, coordinated with state and federal health officials, as well as investigators across other affected regions in Brazil, to harmonize study protocols and initiate a series of groundbreaking epidemiological investigations that connected the microcephaly cases to Zika virus infections during pregnancy, ruled out alternative hypotheses for the causes of the epidemic, including larvicides, and provided the first clinical descriptions of a new disease, congenital Zika syndrome. What had previously been thought of as a benign mosquito-borne virus was soon recognized as a global threat capable of causing severe teratogenic effects. Building on this initial body of research and with key support from the Zika Plan Consortium, our team of investigators has initiated and followed up a series of clinical cohorts across Brazil to study Zika virus infections in pregnancy. The Zika Plan affiliated cohorts include the Microcephaly Epidemic Research Group's Pregnant Women Cohort and Pediatric Cohort in Pernambuco State, the Zika IFF cohort led by Professor Maria Elizabeth Lopez Marrera in Rio de Janeiro, and also a pregnancy and birth cohort in Goya State led by Professor Marilia Dalva Turki. Our Zika Plan affiliated research has had three primary areas of focus. Our first area of focus was to shed light on Zika virus infections in pregnancy. In particular, our research has contributed to the development of a diagnostic algorithm for identifying maternal infections, yielded new insights regarding the vertical transmission of Zika virus, and provided some of the most robust estimates to date on the absolute risks associated with congenital Zika virus infections. Our second area of research has been on the prognosis and health needs of children with congenital Zika syndrome. Specifically, we have worked to understand the full spectrum of the disease, characterizing the structural abnormalities, functional impairments, and neurodevelopment of children with congenital Zika syndrome. And our third strand of research, which was led by my colleague, Professor Hannah Cooper at LSHTM, focused on the social and economic impacts of congenital Zika syndrome. So in addition to its direct impact on the health of congenitally infected children, Zika virus infections during pregnancy can have profound implications for the well-being of mothers and other caregivers. And this vital social science research has highlighted challenges related to support systems, gender inequalities, maternal mental health, and healthcare access. In addition to collaborating within the Zika Plan Consortium, our team of investigators has been actively engaged with the global Zika research community. And one notable initiative is the Zika Brazilian Cohorts Consortium, which is led by the Microcephaly Epidemic Research Group in partnership with the Brazilian Ministry of Health. And the ZBZ Consortium brings together pregnancy and birth cohorts of children with congenital Zika syndrome from across the country of Brazil in order to facilitate large-scale joint analyses of the data. In addition, the data that we've generated through our clinical cohorts has fed into other large-scale individual participant data meta-analyses, including an initiative through the European Commission Vertical Transmission Study Group with our partner consortia Zika Alliance and Zika Action, and also the WHO-led Zika Virus Individual Participant Data Consortium. And I just wanted to briefly highlight a few of the really key outputs from this work. And the first one is to note the MERG pediatric cohort, which has now been followed up for 5 years, is the largest single cohort of children with congenital Zika syndrome and Zika-related microcephaly. Another key output is the scientific articles that have been produced by studying these cohorts, which include more than 40 publications with another 15 in preparation. And finally, we've also done a lot of work around expanding the efforts and thinking about epidemic preparedness through research capacity strengthening, including with the training of more than 30 master's students, PhD students, and postdoctoral scientists across our institution, and also being involved in epidemic response training internationally through MOOCs and other outbreak simulations. Our research has also fed into policy, in particular through guidance of the Brazilian Ministry of Health and the WHO PAHO. And with that, I want to introduce our speakers for today. And so our first speaker will be Professor Maria Elizabeth Lopez Marrera, who is a Fiocruz researcher specializing in pediatrics, child growth, and neurodevelopment. After that, we'll be joined by Ricardo Arias de Alencar Jimenez and Democrat de Barros Miranda Filho from the Microcephaly Epidemic Research Group. So thank you very much. There are many amazing scientists behind this work, but I'm really glad that we'll get to hear from 3 of them today. So with that, I'll pass over to Professor Marrera.
And first of all, thanks for inviting me to present our results. You have 5 papers published about the neurodevelopment of these children. And I put in the last slide, but I would like to begin to thank all the people that help us to bring these children to visibility, because they usually be invisible. And just now, they are not in spotlight anymore, and they are still having problems, and they still needs to very hard follow up in order to bring to them some hope and some clinical results. This is results of a cohort. Then these cohorts are registered in the clinicaltrials.gov. We enrolled 296 children, and 96 of them had microcephaly at birth of any other complication as calcification or seizures or arthrogryposis, something like that. And you have another children without problems at birth. They was born completely with the clinical normal evaluation. And nowadays you have 146 children in follow-up, and you already 17 deaths. Of course, all of them are in the group of microcephalic children. And during the follow-up, we did some measurements of gross antibody composition, neurodevelopment using Bailey tests 3rd edition, high and hearing exams, and clinical evaluations. This is the 2 more important publications that you have, one in the Nature and another one in the New England. And one of them is the children with 1 year old, and another one 2 years old. And you continue to follow these babies, and you want to follow them from now until the school age. And our result is very worried, because you note that these babies that was born apparently normal, they used to have some delay in development. Most of them, around 30%, had some delay in language domain in the Bailey tests. Some of them had some cognitive and motor delay, but mainly they have language delay in the language domain. This is very important, because you know that follow this, the language delay, you can have the cognitive delay. Then they are arriving the school age. Then that's very important to define a risk and to send them to have some intervention in order to minimize their problems, their impairments in the school age. Then the results about the neurodevelopment are language delay, especially receptive language, was found in infants prenatal exposed to Zika virus and asymptomatic at birth, which might indicate future learning difficulties. Neurodevelopmental dysfunction, mainly in the language domain, was unrelated to the gestational period of exposure. Because you know that microcephaly used to be more usual in the mothers, in the babies from mothers that have the Zika virus in the first trimester of gestational age. But this type of language delay is not related to gestational period of exposure. The exposure to Zika virus in pregnancy can affect the neurodevelopment of children who were asymptomatic at birth. You also noted that the head circumference of these babies with delay is smaller than in the babies without any delays. They are not microcephaly, but they have a smaller head circumference at birth for gestational age. In conclusion, a normal assessment at birth after congenital Zika virus exposure does not guarantee ongoing normal neurodevelopment in these children. Exposed infants should have regular development of ophthalmological and hearing evaluations, even if initial exams or evaluation are normal. Early detection of risks can accelerate the referral to children for early intervention. And you know that these children have some neuroplasticity. We strongly believe in neuroplasticity. You can do something using some intervention, neurodevelopment interventions. Understand the full spectrum of neurodevelopment abnormalities has important in public health and education system implications. Then you conclude that these children need to have neurodevelopment assistance because you can identify early risks and send them to have some interventions. And our perspectives from now is continue to follow neurodevelopment of these children from 3 years old to school age. You know that the new test cannot be daily anymore. You need to send these tests for VIPSI tests or other tests. And another thing is to compare neurodevelopment of these exposed children with no-exposed children of the same age. Of course, this is a big problem for us because you are using clinical mother symptoms at pregnancy and serological tests from mother and child just now. And you are going to have PRNT2 from these children. And I hope to have this comparing group. But you know that in the birth cohorts from Brazil, begin before the Zika virus epidemic, you used to have 10% of the delay language domain. Then now you have almost 30%. Then maybe this is the causality of this probably is really the exposure to Zika. But you hope to have this comparing group using child with the same age. And I would like to thank Zika Plan, Wellcome Trust for London School of Hygiene and Tropical Medicine Help, CNPq, FAPERJ, Brazilian Minister of Health, Fiocruz, NIH with RO1. Without them, it was impossible to follow these children neurodevelopment because they are followed because you have assistance in research. Our system doesn't have regular help with these neurodevelopment tests for everyone. And I'm very thanks. I would like to thank a lot of all this financial support to these children. And thanks a lot for this. Then that's it. I would like to finish with this thanks and thanks to my team, all the team that work with us and the Instituto Fernandes Figueira from Fiocruz, Rio de Janeiro. Thanks.
Thank you so much, Vivette. And I see that we do have a question, but we're going to save them until after all three speakers, and then we can answer them together as a panel. And so with that, I'm going to pass along now to our next speaker, who is Professor Ricardo Jimenez, who is a MERG researcher specializing in epidemiology and a professor at the Federal and State Universities of Pernambuco. And Ricardo will be presenting on the risk of adverse outcomes in the first years of life in children born to mothers exposed to Zika virus infection during pregnancy.
So, good morning or good afternoon. And I'd like first to thank Zika Plan for the opportunity to present here the results of the work we have been developing. My talk will be on the risk of adverse outcomes in the first years of life in children born to mothers exposed to Zika virus infection during pregnancy, and I'm talking on behalf of the MERG group. To be able to talk about the risk of adverse outcomes after infection during pregnancy, it was necessary first to establish a definition of what was exposure during pregnancy. So we worked on categorizing these women that had rash and were notified to the health secretariat of the state of Pernambuco during the Zika epidemic. We categorized them either as positive or as suspected. And these two major categories, they included subcategories depending on the results of a molecular test, PCR, and of serological tests. Two markers of recent infection, IgM and IgG3, and one marker of previous infection, PRNT. And these tests were performed sequentially during pregnancy. Here we present the main characteristics of these 501 women that we followed until delivery. So they were young women. The median age was 26 years. Most of them had secondary education. Most of them belonged to a lower social class. Most of them, again, had a previous pregnancy, but the report of children with malformations from previous pregnancy was low. It was only about 3%. The frequency of smoking was less than 10%. The frequency of illicit drug use was less than 2%. And most of these 501 women were categorized as being Zika virus positive. And most of them in this group of positive women were both evidence either based on PCR or based on the serology. From now on, I present the absolute risk of the adverse outcomes of the children born to these mothers. So the absolute risk of microcephaly was around 3%, and it was not different when we consider the women that were categorized as positive or the whole group of women, we mean the positive plus the suspected. Another point that I would like to highlight is that we observed that microcephaly is not a homogeneous entity. In fact, microcephaly itself is on the spectrum, as we observed children with proportionate or disproportionate microcephaly, and with or without imaging, ophthalmological and neurological abnormalities. And these results are consistent with the results of other cohorts in a case control study that we conducted in Pernambuco. Here we presented the risk of image alterations, and the risk of image alterations was about 7%, and it was not different when we consider just the group of positive women and the group of positive plus the group of suspected. Calcification and ventriculomegaly were the most frequent abnormalities that we found, and these abnormalities, they were also very frequent in the case series of microcephaly that were reported in the literature. Of course, in the case series of microcephaly, there is a wider spectrum of image abnormalities in these children. In relation to the neurological abnormalities, the risk of neurological abnormalities, it was around 5%. If the assessment of the children was done, when it was analyzed at the first evaluation, it was around 5%. And when we consider the whole period of follow-up, it raised to 13%. So it's not surprising that there was this increase during the period of follow-up, because some finds like epilepsy, like motor and material milestones may be detected only in a later age of these children. The frequency or the scale of ophthalmological abnormalities was around 7%, and it did not differ between the two groups of mother, and it did not vary if the detection was obtained at the first evaluation or at any time during the follow-up. The risk of abnormal auditory brain stem response was about 5%, and again, it did not differ if we consider any of the two groups of women, and if we consider if it was detected at first evaluation or detected at any time during the follow-up. The risk of dysphagia, reported dysphagia by the mothers, was around 2%, 2 to 3%, and then again, it did not differ if detected at the first evaluation or at any time during the follow-up, and in both groups, in children born to both groups of women. The results of these slides, I think that they are very important because they show that the risk of presenting at least one of the main abnormalities compatible with the congenital Zika syndrome is about 20%, and 20% when detected at first evaluation, and about 30% when detected at any time during the follow-up. This slide also indicates that the individual abnormalities, they are more frequent than the combined abnormalities. And here we selected just the main abnormalities, which are microcephaly, imaging, neurological, and ophthalmological abnormalities. In relation to the combined abnormalities, the frequency that we observed, it was around or less than 2%. So the risk of presenting microcephaly and neurological abnormality associated with imaging or ophthalmological abnormality was below 2%, as well as the risk of presenting neurological abnormality associated with imaging or ophthalmological abnormality. And these figures were similar when we consider the whole period of follow-up, and when we consider the group of children born to mothers who were positive, and also the group of mothers who were suspected of Zika virus infection. So the main conclusions of our results are that the frequency of microcephaly was approximately 3%, so it is relatively low, and it is consistent with the finding of other studies who found frequency of microcephaly of less than 6%. Another important finding of our work is that the risk of presenting at least one abnormality compatible with congenital Zika syndrome was around 20%. Our findings lie between the study of Brazil, who found a frequency of 50%, and the studies of Reynolds and Roen, who found a frequency or a risk of about 10%. Another important finding is that isolated manifestations occur more frequently than in combination, and in fact, that combined adverse outcomes, they occur in less than 3% of the children born to mothers who had Zika virus infection during pregnancy. And using the classification of exposure, we were able to estimate the risk of adverse outcomes according to the degree of evidence of Zika virus infection during pregnancy. So I would like to thank Zika Plan for the partnership and for the support. And I'd like also to thank the other supporters of the work that the Marrera has been developing. Thank you.
Thanks very much, Ricardo. And our next speaker will be Professor Democrito Tabaro Miranda Filho, who is a MERG researcher specializing in infectious diseases and a professor in the Medical Sciences and Health Sciences postgraduate program of the University of Pernambuco. Democrito will be presenting on the contributions of the Microcephaly Epidemic Research Group pediatric cohort to the understanding of congenital Zika syndrome spectrum.
I'm very grateful to be here to present our contributions to the understanding of the congenital Zika syndrome spectrum. In 2016, we published these 4 papers that contributed to an initial description of microcephaly. In a large case series, these 104 patients. And we also proposed an initial description of, at that time, a presumed congenital Zika syndrome. And we showed other less typical findings, such as microcephaly without calcifications or with normal brain imaging and intrauterine growth restriction in our case control study. So now I will show what else we have done after the Zika Plan kickoff meeting here in Recife in October 2016. So this is our cohort. It is composed by 4 groups of children recruited in different situations. These 4 groups. These, with almost 200, have been followed since the beginning of the epidemics, and almost all of them have with microcephaly, are outpatient groups. And this other group have 80 participants are from our case control study. 34 were cases with microcephaly at birth, and the others are control, not neurotypical children. This third group are born to mothers who had rash during pregnancy that composed the married pregnant women cohort that Professor Jimenez presented before. So in this figure resumes part of the results of this article in which 91 children with Zika-related microcephaly were sequentially evaluated for epilepsy, and the cumulative incidence of epilepsy that we found in the first 2 years of life was about 71%. And the most common type of seizure was spasm epileptic without hypsarrhythmia. These are the most important. In this paper, we show the frequency of oropharyngeal dysphagia in children with Zika-related microcephaly. It was almost 80%. And in children born to mothers who had PCR positive for Zika during pregnancy, it was 80.6%. And this frequency is higher than expected for healthy population in general. This was a study to evaluate neurodevelopment with a screening test this week in 4 groups of children, these 4 groups here. And what we found that was almost 100% of the children with severe microcephaly were classified at risk for retardation according to the test. And there was also a high frequency of children at risk among those with moderate microcephaly. And the risk of retardation in this group that was only exposed during pregnancy but without microcephaly was similar to the neurotypical control groups. Other component of congenital Zika syndrome we recently identified was cryptococcidism in children with Zika-related microcephaly. From 22 males examined, 8 had cryptococcidism, and about 80% of the testes were palped in the inguinal region. Other 7 boys that were not included in this case series had retractile testes. This is another kind of finding is also named the false cryptococcidism, because you can put temporarily the testes in the scrotum, but after that they were retractile to inguinal region. The most important findings in surgical findings of that series were a high prevalence of testis epididymis dissociation, showed here and here, a low mean of testicular volume for their ages, so like hypertrophy. And another one, it was ectopic gubernacular insertion in all cases. All cases had a gubernacular insertion that suggests that these may be explain the mechanism of cryptococcidism. Other recent finding is a relatively high frequency and early presentation of adenoid hypertrophy in children with also Zika-related microcephaly. It was about 54% of those with symptoms of respiratory obstruction that were examined. We found adenoid hypertrophy very severe. 13 of them were submitted to surgery, and in 11, there was complete resolution of symptoms. And finally, we recently also described the finding of different types of endocrine dysfunction in 30 children with Zika-related microcephaly. This is the first study in the literature to discuss endocrine disorders, and the main dysfunctions that we found were variants of early puberty, or pubarch or adenarch, short stature, hypothyroidism, obesity, or these kind of different combinations that we show here. So our conclusions are there is a spectrum of severity in the Zika-related microcephaly, and the congenital Zika syndrome is not yet totally understood. In addition to damage to the central nervous system and its direct consequences, such as epilepsy and mental retardation, children with severe Zika-related microcephaly may present other anatomical or functional impairments. Some of these impairments are dysphagia, cryptococcidism, retractile testes, adenoid hypertrophy, and endocrine dysfunctions. So next steps is necessary to say that children known to be affected, as well as asymptomatic children at birth or in their first years of life, must be systematically monitored in order to identify dysfunctions or late manifestations or development problems. So we have to thanks to the Zika Plan Fund and the other supporters of our groups and all our collaborators and camp field workers. Thank you.
Thanks very much, Makoto. And could I invite Bebet and Ricardo to join us as well? And now we'll have an opportunity to answer any questions that you might have. And for our audience members, if you could please put those into the Q&A function, we'll start off with those questions. Great. Well, thank you so much, everyone. That was really amazing and an incredible research that's been done over the last 5 years in describing this new disease and learning about how the vertical transmission of Zika infections in pregnancy work. The first question, I think, is probably going to Professor Marrera, and it's a question about the children who were born apparently asymptomatic at birth. And do you have a sense of what the estimated proportion is of the children who we know that they were exposed to Zika during pregnancy, but then were born without any apparent neurological complications?
Around 70% doesn't have anything. They was born normal and they have the neurodevelopment as completely normal from birth to 3 years old. Then that's a good note. This is a good news for the mothers, but we think that you need to follow them at least to school age in order to detect the risks early, and maybe if they need to send them on some interventions.
Great, and I think that's a good follow-up point as well, is that you had mentioned that the children are likely to have a great deal of neuroplasticity, and so there's having the neurological interventions and stimulation for the children is very important. And perhaps Democrito, you might want to weigh in this as well. But what sort of interventions are being shown to be the most effective for these children?
Yes, we are doing the speech language interventions using some programs with them, and physiotherapists intervention, because you know that sometimes you just need to correct the posture and they improve a lot. And the Juntos program, I don't know if, I think that you know about that. This is a program with Hannah Cooper and Loveday and some Tracy Smith. And we introduced this program here in Brazil, but of course you are using this program to children with the high risk for cerebral palsy, especially the microcephalic babies. And this program is going very well, and the mothers are very satisfied with the results. And the fathers used to go to see the classes too. But for the other children, these children that was born apparently normal, we don't have a specific program. You need to detect the problem in order to send them to have some intervention. In our population here in Brazil, the results from birth cohorts from Pelotas and Ribeirão Preto, Brazil, they usually show the 10% of the delays in language. And of course, you are seeing that the higher delay in language in the Zika exposure to children. But of course, the idea is to have a control group. In our country, you are programming this, but it's almost impossible because you don't have good serological tests until now. And to choose some children that was born in the epidemic period of time and at the same time could be almost impossible. But just now we are trying to do the IgM and the IgG in these children, in the mothers, and try to have some babies that you can say that they didn't have exposure to Zika, and try to follow their neurodevelopment.
Great, thank you. And I think our next question is for Ricardo, and it's a question about whether or not the absolute risks that you described were derived from during or after the Zika outbreak that occurred in Brazil.
Okay, so we started recruiting these women for the cohort of migrant women as early as February 2016, so just after the peak of the microcephaly epidemic. So if we had started before, maybe we would have a larger number of women, but it does not affect the risk. Among those infected, we do not think that the risk would change over time. I'd like to call the attention to one point that people sometimes get a bit confused, is that we are talking about the risk of adverse outcomes. So it's not, it's different than what people are more used to read about, which are the cases of microcephaly. So for the children with microcephaly, it is more frequent, more common to present more than one abnormality. But as already explained, microcephaly is has a spectrum itself. There is a variation in the microcephaly. But more important than this, I'd like to call their attention is that if the child does not present microcephaly, the risk of presenting more than one abnormality is below 3%. So the risk is below 3% of presenting combined abnormalities related to Zika virus infection during pregnancy.
Thank you. And to follow up on that, a question has come in around Zika-related microcephaly in other regions of the world. And I wondered if any of our panelists could weigh in on whether or not you have a sense of whether or not you're also seeing Zika-related microcephaly occurring in other regions of the world, or is it being misdiagnosed?
So we have publications describing Zika-related microcephaly in almost all countries in Latin America and Central America, and with the same characteristics. And the other publications about cohort studies with pregnant women, they show a similar risk of microcephaly in this that occur in Zika virus.
The variation that we found in the clinical presentation of microcephaly in our study is similar to what was described in previous study. In previous studies, it was also found microcephaly with a proportionate or disproportionate proportionate or disproportionate microcephaly with or without imaging, neurological or ophthalmological abnormalities. So it seems maybe some authors did not call the attention to that point, but if you go through their papers, if you read their findings, in these other studies, there was also a variation in the clinical presentation of microcephaly. So it seems that it does not differ from one place to the other.
Thank you. I wanted to pick up now on a point that Gustavo had raised in the really nice opening session, which was talking about the impacts of the COVID-19 pandemic on the children with congenital Zika syndrome and their families. And I wondered if you could weigh in on what you have seen within each of your cohorts. Yeah, Gustavo, it would be great to hear from you as well and what your perspective is.
Okay, I'm sorry. I was thinking that
perfect.
It's just for you. No, it's okay. The impact, I also use it to talk with Maria Elizabeth Marrera, and we are very worried how to support the cohort student without funding. So this is a very important impact on how not just to keep our study, but also to support and give access to the family and children. So I think that this is a very critical issue that we could have to discuss globally, because there's a lot of inequalities to support scientific research in our country at the moment. So to give you an idea, Brazil has cut something about 70% of the science and technology budget from the federal government. So, and now we are living during a COVID pandemic, but we continue to live to give assistance to children with Zika congenital syndrome. And we also have an important dengue and chikungunya epidemic at the moment. So the problem didn't disappear, right?
Yes.
This is a very complex issue that we need to continue to discuss, especially with inequalities within country in Brazil and globally.
And yes, I would like to comment this. You know that these children are not in spotlight anymore. And the children with microcephaly continue to need hospitalizations. They are having seizures. They aren't have the medications that they need to treat the seizures. And you have a discontinuation of these offers to this support. And I really think that the children with microcephaly need this to have more assistance, and maybe they need to come back to spotlight. They can't be out of the spotlight because they really need the assistance. And my worry is about all children that was born in the pandemic period, because you know that 70%, almost 70% of the mothers would have Zika without symptoms. And maybe they can have some delays in development, and they need to be followed up. They need to have adequate follow-up in their development. And you know that without this assistance in research funds, I really don't know how to do it. That's a big question, because 30% of delay in language is very strong, and you need to have some attitude in order to improve this, in order not to have an impairment in the school age.
Thank you very much. And Gustavo, you had your hands up. Do you want to weigh in?
No, very quickly. So just to highlight 2 points, right? One of them is also related to what Maria Elizabeth Marrera, it's our Bebet, said. So we didn't include in the prenatal orientations about Zika prevention. So we are running now a WHO research about risk communication on current pregnant women, and we don't have anything in the prenatal about Zika. Even information, even how to prevent, and also serological tests or some diagnosis tests during pregnancy. So this is very, very critical about after our outbreak. The second point is not just about Zika, but other multi-disabled children in Brazil, because we don't have a good, it's not only in Brazil, but we don't have information system adequate to identify these cases. So we don't have any idea how the amount of people are born with multiple disabled symptoms. So this is very, very critical. And I think that globally we need to discuss this, especially in low-income countries.
Thank you. Yes, Ricardo.
Yeah, I'd like just to make a point of the relevance to continue following these children. I mean, it's very important to study the impact of the COVID-19 on the development of these children, because some achievements that were reached, now these children are losing these achievements because they are not being stimulated properly. It's not possible to stimulate them properly during this period of COVID-19. So it's very important to document and to think of strategies on how to deal with this situation. And I think that it's also very important in relation not only to the children, but also to the mother. I mean, these mothers, they are not working, they are taking care of their children full time, and they also do not have access to the health facilities because of the COVID-19. So there is an important impact on the life on the history of, on the trajectory of these mothers and on the development of the children.
Thank you all. One thing that's really struck me about the Zika response is the scale of collaborations within Brazil and also internationally. It's really remarkable and kind of a template for other emerging infectious diseases in many ways. And I wondered if you could weigh in about what you see as kind of the secrets to success for international collaborations and within country collaborations during emerging infectious disease outbreaks.
This is for me or?
For all of the, any of the panelists. It would be great to hear from you, Gustavo.
Okay. No, just a brief comment. As I show in my presentation, the Brazilian cooperation was very intense, especially with the US and other countries. So, but during the cooperation, we have many new experience to face as data sharing, how to deal with data sharing globally, because when we receive international funding, it's compulsory to share the data. Also, pathogen sharing, it's a very sensitive issue. So how to create a biobank and how we can deal with patents and intellectual property and authorship. So this is also important when Brazil start or intensify international cooperation about after Zika outbreak. And now we use the same network to improve COVID research cooperation. So it was very, very useful, and we learned a lot with the Zika outbreak to respond to COVID research and cooperation internationally.
I think that one very important point is to work very early on harmonizing protocols and harmonizing instruments that will be used to assess the effect of the infection. This is what made it possible to analyze the data together for the Brazilian Consortium and also for the WHO Consortium. And this is a very important step in the beginning of every epidemic, is the harmonization of protocols and of instruments. It's not possible to conduct the multicentric studies during the epidemic, but it's possible to harmonize the data, to harmonize the protocols, and then make the data comparable and analyze them together.
I agree with you, Ricardo and Gustavo. And this is a challenge for us in the Zika times. And now you are, as you learn with Zika, it's a little better with the COVID researches. And you know that I think that you have here in Brazil a biodiversity law that are very difficult to us to share samples with the other countries. And this maybe could contribute with this absence of the serological tests, good serological tests. And this is a big challenge for us to share samples of blood, urine, everything to other countries. Is very bureaucratic. And it's very difficult to work with this type of thing.
And so I just wanted to say thank you so much again to all of our speakers. We really appreciate it. I lost track of time there with the Q&A. It was such an interesting conversation. But now I'll pass over to Annelies. Thank you, everyone.
Welcome to this session entitled New Insights into Zika Virus Biology, which I have the privilege to chair. Let me share my screen now. So in this session, we are going to highlight some of the basic research discoveries that were made in the Zika Plan Consortium. And I'll start with a talk entitled "What Mosquito Genetics Taught Us About Zika Virus Emergence." And one could wonder what mosquito genetics have to do with Zika virus emergence. And in this talk, I'm going to try to illustrate that the two are actually connected. Zika virus was first isolated in 1947 from a sentinel monkey in the Zika forest of Uganda. And for the next 60 years, it was considered an innocuous virus because there were only a few, actually less than 20 reported human cases, and all of them were associated with mild and self-limiting symptoms. In 2007, Zika virus was responsible for a relatively large outbreak on the island of Yap in Micronesia. In the following years, it caused other outbreaks in islands of the Pacific Ocean, namely New Caledonia and French Polynesia, for example. In 2014, Zika virus reached Brazil, where it was introduced and then spread out until it was responsible for a massive outbreak throughout Latin America and the Caribbean. It culminated in 2016 when the World Health Organization raised the status of Zika virus to that of public health emergency of international concern, primarily because of these novel clinical manifestations such as the neurological complications and the birth defects. So in the last few years, considerable research effort has been made to try and understand the factors that may have triggered and facilitated the spectacular emergence of Zika virus in the Pacific and in Latin America. Perhaps less attention has been paid to the reverse situation. There are regions of the world where Zika virus did not emerge. And one of those regions is Africa, where we know the virus is present since it was isolated there for the first time. And it has been circulating in the human population, as was seen in Angola in 2016 and previously in Gabon in 2007. So it is kind of puzzling why there was a lack of large-scale human outbreaks in Africa, despite seemingly favorable conditions. There is one single exception, which is Cape Verde, where massive outbreaks occurred in 2015 and 2016. What we hypothesized was that perhaps this exception to Zika virus emergence in Africa may have something to do with this mosquito, Aedes aegypti. The reason why it could have something to do with this mosquito is that Aedes aegypti consists of two subspecies. Aedes aegypti formosus, the African subspecies, which is a dark form primarily found in sub-Saharan Africa, and Aedes aegypti, the globally invasive subspecies, which is found in the tropics and subtropics of the rest of the world, in Asia and in the Americas. Aedes aegypti formosus is a generalist that breeds in a variety of urban and forest habitats, whereas Aedes aegypti is a human specialist that preferentially bites humans and breeds in the domestic environment. Aedes aegypti, the human specialist, is thought to have evolved from generalist ancestors around 5,000 to 10,000 years ago, somewhere in Western Africa. And its domestication allowed its global expansion during the slave trading period. To experimentally test whether the two subspecies of Aedes aegypti may have different abilities to transmit Zika virus, we sampled populations that were representative of the two subspecies around the world. 3 populations from the Americas, 3 populations from Africa, and 2 populations from Southeast Asia. And then we exposed these mosquitoes to a panel of 6 Zika virus strains representative of the currently circulating viral genetic diversity. And the results of our survey are represented here on this slide. You can see pie charts, which each represent mosquito population. Each slice of the pie chart is one of the 6 viruses of our panel. When the color is blue, it means that the mosquito is susceptible, when the color is red, it means that the mosquito is resistant. So what you can clearly see already is that the 3 mosquito populations from Africa are more red than the population from out of Africa, which are more in the blues, which means that mosquitoes from Africa are more resistant on average to Zika virus infection than mosquitoes from the rest of the world. And so it looks like Aedes aegypti formosus, the African subspecies, is more resistant to Zika virus infection, regardless of the virus strain, than the Aedes aegypti globally invasive subspecies. To further investigate the genetic basis of this difference, we used 2 complementary approaches. In the first, we took advantage of natural hybrids between the 2 subspecies that occur in some places of Africa, and we measured the relationship between Zika virus susceptibility, which is represented here as the oral infectious dose 50%, which is the dose of virus in the blood meal that is required to infect 50% of the mosquito population, as a function of the proportion of the Aedes aegypti genome relative to the Aedes aegypti formosus genome in those hybrid populations. And you see that there is a negative relationship, meaning that susceptibility increases as the proportion of Aedes aegypti genome increases. The other approach that we use is to perform an artificial cross between mosquitoes from Guadeloupe, representing the Aedes aegypti subspecies, and mosquitoes from Gabon, representing the Aedes aegypti formosus subspecies. This cross allowed us to perform genetic mapping and locate genetic factors on the 2nd chromosome of the mosquito, which are responsible for the difference in Zika virus susceptibility between the 2 mosquito subspecies. In conclusion, our findings indicate that the domestication of Aedes aegypti and its subsequent global spread out of Africa was associated with an increase in its susceptibility to Zika virus infection. And we think that this may have facilitated Zika virus emergence, not only through increased vector-host contact, but also due to the enhanced vector susceptibility. This, we believe, may offer an explanation for the lack of large-scale Zika virus outbreaks in Africa, where the mosquito subspecies that is dominant is more resistant to Zika virus infection and subsequent transmission. With that, I would like to thank all of the people who contributed to this study, particularly Fabien Aubry, a talented postdoc who led the work that I presented today. And thank you for your attention. Now I'm going to stop sharing my screen and turn it over to the next speaker. So again, we'll do a session of questions and answers at the end of the session, after the 3 speakers of the session. So the next speaker is Daniela Weiskopf, who is an assistant professor at La Jolla Institute for Immunology in California. Daniela is one of the world experts in T-cell immunity against viruses. And today she's going to talk about pre-existing T-cell memory against Zika virus, of course. Thank you, Daniela. Over to you.
Thank you so much, Louis, and thank you for your interesting talk. And I think this session is so great because this session looks at the difference in virology, immunology, and also other mouse models from all sides of it. The question that I was most interested in, if there is a pre-existing T-cell memory against Zika in people that have never eaten it. Basically, in other words, I was wondering, can exposure to one virus influence the subsequent infection with a different virus? And That question is all based on the hallmark of the immune system. So T cells are part of the adaptive immune system, and the hallmark of the immune system, the adaptive immune system, is that it actually can remember. So once you have an infection, a primary infection, you will, after clearance of the virus, form memory T cells, and these memory T cells linger around and stay with you for like, years, or many, also for a lifetime. So the question is, what happens when you see the same virus again, or if you see a virus that is closely related to it, so that you can assume there's some cross-reactive reactivity. So there's different scenarios. So you can imagine if you see the same virus again, and that's what is basically the premise of subsequent infection with the homotypic virus. You have protection and you have the viral clearance. What happens if the secondary virus is a heterologous virus? So it's another dengue serotype, or in this case, a Zika infection. So you can imagine you can either have cross-reactive protection and viral clearance, or you have insufficient cross-protection and contribution to pathogenesis. So that's all of the different scenarios that can happen if you have subsequent infection with a closely related virus. And why are we interested in that in the case of Zika, which is a very graph here to show you, there is a wide geographical overlap in these flavivirus distributions. So all of the areas where Zika has been circulating is areas where dengue virus is circulating before. And also other viruses do have, other flaviviruses have overlapping areas of this. So the question is, these are all closely related viruses. Do infection, any of these viruses have an effect of subsequent infection? And that's the question we wanted to ask. And particularly in this case, we wanted to ask areas that have been exposed to dengue virus before, which is, as I said, many of these endemic areas where then Zika was circulating, have any influence of Zika virus. So in order to answer these questions, you need to know which exact part of the virus are recognized by T cells. And in contrast to like B cells and antibodies, T cells do not recognize the entire virus. They recognize small parts of it that are presented on the surface of infected cells. So in order to know which are the ones that are recognized, we actually had to map these responses. And this is just to show you a quick assay strategy. So we always are interested in the entirety of the virus. So this shows you that we mapped pools containing many peptides, 20 peptides each, spanning the entire virus. And once we realized which one is positive, we then deconvoluted it to find the exact sequence of the virus that is recognized. And in this case, this was all tested in donors that have been exposed to dengue virus, but have been collected way before Zika virus was circulating. So we're very sure that these have not been exposed to Zika virus, but have been exposed to dengue virus. And this is just an example of how we measure this. So in case you see a positive response, your ELISpot spot assay lights up, like T cells make interferon gamma responses against these peptides. So this helps us to identify exactly which one of these areas of the virus are recognized. So if you now look at the results overall, this is the summary of the epitopes per donor and also the magnitude of the positive responses that we have identified. It's quite remarkable to see that out of all the donors we tested, on average, every donor recognizes 5 different areas of the Zika virus, even though these people have never seen a Zika virus. There's certain donors that recognized close to 40, but that's certainly the exception. So overall, every donor sees about 5 regions of the Zika virus without having seen the virus itself. And if you see in magnitude, you can also see quite a different spread of it. There is certain areas that are recognized with high magnitude. That means the T cells recognize it with a response of a lot of interferon gamma. And there's definitely certain areas that are recognized lower, but still all account for a positive response against it. So if you look into more detail on where these areas are that are recognized from the Zika virus, this is just a summary to show you where all of the different proteins account for the recognition. And you can quickly see that we found cross-reactive epitopes in all of the proteins that we were looking at, with the exception of NS2B, which is some protein we did not find any of the epitopes. But other than that, we found epitopes in all of the Zika virus proteome, in case though with different responses, as I just mentioned before. Like some are recognized very strongly, some are recognized rather lowly, for example, this one NS2A. So this is a different way to look at this. This is the epitopes per protein, and this is the magnitude of response. And you can fairly quickly see that the strongest responses and also the majority of responses is against proteins that are conserved between flaviviruses. We know NS3 and NS5 are highly conserved between not only the different serotypes of dengue, but also between dengue and Zika virus. So not surprisingly, this is where we found the majority of the responses, cross-reactive responses. So if you then look into this different area, different way of plotting the results. So if you plot the magnitude of the response based as a function of the similarity between the viruses, so this is the sequence identity between the Zika virus and the dengue virus, you see that the strongest ones are actually recognized in the highest conserved areas. So this is, we were interested then into actually into more detail to what is the exactly magnitude of the responses that are recognized. So we made T-cell lines with all of these epitopes, and keep in mind these are Zika-derived epitopes recognized in dengue exposed donors. So if you then look into more detail, you can see that if you make T-cell lines against the original Zika epitope that we have identified, which is in case of here plotted in the black line, you see that also other serotypes of dengue, so the homologous sequence in other dengue viruses, dengue serotypes, are recognized in this case. We also found cases where like not only other peptides from dengue virus are recognized, but also yellow fever, Japan encephalitis, West Nile virus, and tick-borne encephalitis. So this is definitely an area stemming from the NS3 protein, where not only the Zika protein, the Zika peptide is recognized, but all cross flaviviruses that are recognized. And here you can see there's cases where, like, you know, again, the serodengue serocomplex is mostly recognized and the Zika peptide. So we do see not only cross-reactivity here confirmed on a single epitope level between dengue and Zika virus, but some cases also across the dengue serotype. So that is something that I think is interesting to keep in mind. You cannot look at one infection of a virus as a single entity. All of the people, many of the people living in these endemic areas have been exposed to many of these flaviviruses. So there is effect of previous infection to subsequent infection. That is something that I think we should keep in mind, not only for this case in the flaviviruses, but we have also recently shown that for SARS-CoV-2, where also previous exposure to human coronaviruses can have an influence in subsequent infection. So just to summarize and conclude this part here, we do detect responses against Zika virus in people that have never seen it. On average, 5 different areas of the virus are recognized. The positive response is correlated with the protein size and also the sequence conservation. And the more conserved these proteins are between the different viruses, the higher the responses was. And we showed you direct evidence of cross-reactive T-cell responses when you do the T-cell lines, and you can see that they not only recognize dengue, but also far across other flaviviruses. And with that, I hope I convinced you that that you cannot look at the entity as a single infection, but you always need to keep in mind that people have throughout their lifetime exposed multiple viruses. And I'd just like to thank the great Zika Plan Consortium that has been funding a lot of this research, and shown here our great partners within the network that have been helping collecting the donors in these areas. So thank you very much, and I give it back to Louis.
Many thanks, Daniela. It was a very clear presentation. We're now switching to the next speaker, who is Suzanne Cattain. Suzanne is a group leader at the Rega Institute for Medical Research at the Catholic University in Leuven, Belgium. And she's actively involved in several research programs to develop experimental models to study viruses and develop antivirals. And today she's going to talk about mouse models and the development of antivirals against Zika virus. Suzanne, the floor is yours.
Thank you, Louis. So good afternoon, everybody. So indeed, like Louis said, I will be presenting data obtained in Zika studies using different mouse models. So already 2 years before the Zika virus outbreak in Brazil, we established a model for Zika virus infections in mice to be able to assess the in vivo efficacy of Zika virus inhibitors. And for this, we use immunocompromised AG129 mice. And these lack the receptors for interferon alpha, beta, and gamma, which makes them highly susceptible to Zika virus infections, as you can also see in this graph. So if you infect them with a low inoculum, you will see that this already results in development of disease, and also mice had to be euthanized. If you give more virus to these animals, of course, you get a faster progression of the disease, and mice also had to be euthanized earlier. At the time that we euthanized the mice, we could also detect Zika in different tissues, like in the spinal cord, the brain, but also liver, kidney, and spleen. So this is a very good model to test antivirals. However, although we did screen over 600,000 compounds, we did not identify a good compound with also good pharmacokinetic properties, which justifies, of course, efficacy studies in animals. But we also have a group of people working in the group of Johannites that are working on a vaccine, and they developed a vaccine against Zika, which is actually a chimeric vaccine, as you can see here. So it's actually based on the yellow fever vaccine. And when you vaccinate these AG129 mice, they are fully protected against a lethal challenge with Zika, as you can see here, this blue line. And also viral RNA levels were below the level of quantification, as presented here by the blue squares. We also assessed the efficacy of this vaccine in the intraplacental Zika virus challenge mouse model. And this was established at the University of Liège. And in this model, pregnant mice, which are immunocompetent mice, are infected at the fetal site of the placenta, which results in brain abnormalities in fetuses. Now, when you then immunize these pregnant mice prior to infection, you will see that the viral RNA levels in the brain of fetuses at embryonic day 80.5, which is just before birth, are again around the level of quantification. And we also looked at immunostaining of fetal brains at the same time point. And then you see there is no brain abnormalities detected in these fetuses, whereas fetuses that were born to non-vaccinated mothers are positive for Zika, as indicated by these red dots, and they are also positive for apoptosis, as indicated by the green dots. So it shows that the vaccine is highly efficacious in protecting mice. Now, in addition to testing antivirals and a vaccine, we also use this AG129 mouse model to assess the pathogenic potential of Zika virus strains from both the African and Asian lineage. And this is a collaboration with the lab of Louis Lambrecht here heading the session, and also the group of Laurent Nguyen in Liège. And we assembled a set of 7 recently isolated Zika virus strains, based actually on the broad phylogenetic coverage, the worldwide geographical distribution, and also the minimal passage history. We decided to select 3 non-epidemic, 2 epidemic strains from the Asian lineage, and also to low passage isolates from the African lineage, as shown here. Now, when we infected mice, the H1N9 mice, we saw that all mice infected with the African strains, they became morbid and had to be euthanized on day 6 post-infection. The Thailand strain, on the other hand, showed a considerably more attenuated profile, and we even had 50% survival. And then all the other Asian Zika virus strain, they showed a more intermediate pathogenic profile, as observed here. We also looked at viral load in blood and tissues. And for this, we used a smaller selection of the Zika virus strains. So we tested the 2 strains from Africa, and also the attenuated Thailand strain, and we selected French Polynesian strain as a representative of all the other Asian Zika virus strains. Well, viral load in plasma and in tissues were considerably higher, as you can see, for mice that were infected with the African strain, as observed here by these red and orange lines, and also the red and orange dots. So this clearly indicates that the African strains are more pathogenic and result in a significantly higher morbidity and mortality in these H1N9 mice. We also evaluated Zika virus-induced microcephaly caused by intraplacental injection of the Zika virus in the mouse embryos. For this, we selected 3 Zika virus strains. So we have the Senegal strain from 2015, the Thailand strain, and the French Polynesian strain. At embryonic day 14.5, which is 4 days after infection, embryos infected with the Senegal strain had higher viral loads in different tissues, as you can see here by the red dots. And in addition, all embryos infected with the Senegal strain, oops, wrong button, my apologies, had edema, subcutaneous edema, as you can see in this picture. We also looked at a later time point at embryonic day 80.5, which is just before birth. And interestingly, by this day, the Senegal strain caused massive embryonic death, so we cannot even examine their brains. Interestingly, when we compare the 2 Asian strains, the Thailand and the French Polynesian strain, we noticed or observed that the Thailand strain resulted in a more pronounced microcephaly, and also, you can see that here very clear, larger ventricles than the French Polynesian strain. So together, these results show that the Zika virus strains with higher levels of infection at early during pregnancy are also associated with more severe serotypes later during pregnancy. And also the results from this intraplacental Zika virus challenge model also raises the hypothesis that Zika virus may have evolved towards attenuation by causing birth defects rather than fetal loss. Okay, with this I want to close the presentation. I just want to thank all the people that contributed to this research. Thank you.
Thanks, Suzanne, for this very interesting presentation. We now have about 15 minutes for a Q&A session. So again, let me remind everybody, if you would like to ask a question, please use the Q&A function in Zoom. I'm gonna open the window and read the first 2 questions that were already deposited. Well, I guess the first one is for me. So the first question is from Adam Neira, who's challenging my hypothesis about Zika virus. And so the statement is that a mutation in old Zika happened in Northeast Brazil sometime between August 2014 and December 2014. Well, I'm afraid there is a bit of a misunderstanding here because our hypothesis, at least the hypothesis that I presented today, relates to the mosquito genetic diversity, not the virus. It is definitely possible that Zika virus evolution may have somehow played a role in its emergence. And there are a handful of mutations that have been identified that could have indeed enhanced transmissibility and or virulence of Zika virus after its emergence. But this is not mutually exclusive with the hypothesis I described earlier, which has to do with genetic differences in the mosquito, not the virus. So what we hypothesized was that differences between the African subspecies and the globally invasive subspecies Aedes aegypti could explain the pattern of Zika virus emergence on a continental basis, namely in Africa, where we didn't see any large-scale Zika virus emergence events, as opposed to in South America, the Caribbean, and the Pacific, where there was massive emergence of Zika virus. So our hypothesis is that mosquitoes from Africa were less able to transmit Zika virus, which we demonstrate with a panel of different virus strains. And the result that African mosquitoes are less capable of transmitting Zika virus was correct for all of the virus strains that we tested. And those including not only pre-epidemic Zika virus strains from the Asian lineage, epidemic virus strains from the Asian lineage, but also representative of the African lineage. So again, whether Zika virus evolved is not mutually exclusive with the fact that mosquitoes have different abilities to transmit depending on their genetic background. So I hope this is clearer now. The next question I would like to answer is from Selina Maria Turci Mortelli, and this is a question to Daniela. Daniela, the question is whether the lab results you presented have been described by field studies. So I guess the question is whether there are any field-based studies that support the evidence you presented from your lab experiments.
Yeah, that's a very good question, and that's something that needs to be addressed in the bigger context of epidemiologists. So what we have been showing is that your immune system, your T cells do recognize parts of the virus, even though they have never seen the virus. Now the question is, what does it all mean? And these have been like collected from the field, human studies that have been naturally infected with like dengue virus. So now we need to find out, what does it all mean? Like, does it, having these cross-reactive responses, does it help you, hurt you, or it doesn't matter at all? And in the case of like Zika infection after dengue infection, there has been some studies. One I can think of is epidemiological study coming out of Nicaragua, where they have been showing that if you have had a dengue infection in the last 6 to 8 years before you have seen Zika, you're actually better off than when you have like had dengue infection like from farther out. What we don't know yet, and what is a question I think that's on everybody's mind, so now a lot of these people have seen Zika. What happens if they now see dengue again? And that's another question that is coming up right now, because there is a lot of dengue infection now in these same areas. So I think that is something that will need to be addressed with larger epidemiological studies, where they collect all this information on, you know, these samples. So, but it's definitely a very relevant question.
Thanks. Let me follow up on that aspect of interference between pre-existing dengue virus immunity and Zika virus infection. I think, at least that's what I understand, that there is some contrasted results, or sometimes even contradictory results about whether pre-existing immunity against dengue virus or Zika virus actually would protect against dengue disease. It seems that it depends on the serotype or the strain. So I'd like to know your take on this, Daniela.
Yeah, definitely. And the fact is that there's a lot of things we don't know. So what I've been looking at is like the T cell response. It's the very same can be true for like, antibody response that also can have cross, which is something that I have not looked into it. We certainly have shown that it very much depends if you had primary infection. So if you only seen one of the serotypes, you have a different response than when you have seen already like multiple, like saying you had multiple dengue infections before Zika, that induces a more cross-reactive response. So I think there's a lot of different parameters that need to be looked at. And also what I'm thinking is very interesting that it's not like one epitope, it's like already every person recognizes multiple. So I think you really need to look into this in a more global perspective and then see the difference in epidemiological field size. I think just in a lab, it's gonna be difficult to make the connection with what is protective. We can just show like what is recognized, and then field studies need to be shown like, if that has a protective effect or not. Of course, difficult to do, and this is a great network. So I hope we can continue in, having access to samples where we have associated outcome. So we know if people have been infected again and have been protected.
Thank you. I hope so too. We have another question from Abdul Ali Mohammed Lawal, who is making a comment, I think more than a question. African Zika virus strain is more pathogenic, but the vector is less transmissible. So if this is true, the vaccine, which is found to be efficacious, needs to be improved before the pandemic. And it's true that on the one hand, we have mosquitoes in Africa that are less competent for Zika virus transmission, but the Zika virus strains in Africa, those from the African lineage, which relates to the data Suzanne presented, seems to be more transmissible. So it looks like so far, the lower competence of the mosquitoes won over the higher transmissibility of the viruses there. But it is definitely something we need to keep in mind, that those viruses in Africa have potentially more severity and higher transmissibility in the case they escaped from Africa. So I think this is where the comment is headed. And perhaps we could elaborate on that a little bit, if you will, Daniela and Suzanne. What about your thoughts on whether genetic diversity of the viruses, and specifically the existence of the African lineage, which has never been involved in any human outbreak so far, any reported human outbreak, whether this could represent a serious threat to any vaccine and or antiviral in the future?
It's difficult to say, of course. I have to be honest that for our vaccine candidates, we only so far tested it against the Asian Zika virus strain. So this is the MR766, which is of course more kind of a laboratory strain, but we also tested against a strain from Suriname, which is again an Asian lineage strain. So no, I do not know whether it is efficacious against the African lineage strain. So it's possible that it's not efficacious, but yeah, I also don't see why it's not efficacious. But maybe Daniela can better comment on that.
Yeah, I can definitely my view on from the T cell side. So one thing that is good about the T cells is that they recognize multiple targets. So there's never reliance on just one. So that's good news. So even if you have like a mutation in between the lineages in one of the epitopes, you also recognize other ones. I showed you like on average, people recognize five different epitopes. And I also showed you that they not only recognize without the serotype, above the serotype complex, but also like even completely other flaviviruses. So T cells are very tolerant in terms of like mutations. So I would be surprised if that the different lineage would like, take into account. I cannot speak, of course, it could be if you change conformational epitopes, you'd have a difference in antibodies, but on the T cell side, I do think that the response is broad enough that would not make any effect.
Thanks for this hopeful response. I don't see any more questions in the Q&A function. Well, yes, there's one that just popped up. Again from Selena. Is there a Zika vaccine in the horizon? And what would be the limitations of it? I guess that's a question for the two of you, or maybe Daniela first.
I definitely know there are certain companies that are in a development phase of a Zika vaccine. I think it has been somewhat halted because you always need to have efficacy trials. So and we all know that the transmission of Zika virus went down. So many of these companies did not have enough infections to actually prove efficacy of their vaccine. And I don't know if we should hope for like picking Zika infections back up, or we should just be good for now. But I do know that there is definitely vaccines in the work, and they will be having efficacy trials if the virus picks up again.
Suzanne, is there anything you want to add?
Yeah, there it is. The only, because the question is also about limitations. So one limitation could also be, of course, it should also be safe to use in pregnant women, of course, and that's always difficult to test this in the field. But yeah, that I do not have anything else to comment.
Another question from Michael Paul, who's wondering whether the African Zika virus strain explains the difficulty of isolating Zika virus among African countries in many studies. Well, I don't think there is any issue from the virological point of view, because these strains from the African lineage grow very well in cell culture, and they can be isolated from clinical isolates, provided the clinical isolate contains the virus. The reason why we hardly isolate them in the field is because they don't circulate among humans. All of the isolates, or most of the isolates that we have from the African lineage, come from sylvatic mosquitoes, where the virus circulates in its sylvatic cycle. And so the reason why we don't see them very often in studies is because we have only a handful of African virus strains that are available. Most of them are old and heavily passaged, with the exception of a few recent strains from Senegal, which Suzanne showed earlier. But I think there is no technical limitation in isolating these viruses, besides the fact that they're rarely found in humans. With that, I think we're reaching the end of our session. So I'd like to thank the speakers again for the presentations. And then I'm going to hand it over to Annelies.
Well, thank you for the chairs of the two first sections. Elizabeth and you, Louis, you really kept to time. We had very good discussions. I think the topics are absolutely interesting. So we all need a biological break. So we'll now have a break for just 15 minutes. And while you have a break and you have some time and you love tweeting, remember that we have a Twitter account, #ZikaPlanUnite. So do tweet and tell the world what you've learned and about all these interesting talks. So meet you again in exactly 5 minutes, which in the UK is 3:15, in Switzerland is 4:15, and in Brazil, I would not know. So see you soon. So, welcome back. I must say, when I listened to all these fantastic speakers, I thought, what a privilege I've had over the past 5 years to lead this consortium. I mean, real experts, but also, in terms of friendships that we've built over the years, it was just a fantastic experience. So it is now my privilege to introduce James Logan, who is professor at the London School of Hygiene and Tropical Medicine, who will now chair the third session. And it's now from the virus, we are now moving to the vector. Thank you, James.
Thank you very much, Annalise, and hello, everybody. Great to be here, and thanks very much for the opportunity to chair this session. So I'm going to be talking a little bit about, or in the session, we're going to be covering the vector. We heard a little bit about that already from Louis Lambrecht. So we're going to be continuing that theme. Myself, I will be giving a presentation first, and then I'll be followed by my colleague, Kathleen O'Reilly, who will be talking about some modeling work that she has done. So I'm just going to now share my screen and hope this works. Okay, I hope that's working. If it isn't, please somebody do jump in and tell me. So I'm going to be talking a bit about the work that we've done in understanding how we might best protect ourselves against the vectors of Zika, and the work that we've done in terms of understanding people's perceptions and knowledge around this, and how we can support and build capacity in vector control and vector research and preparedness through online resources, but also through mathematical modeling as well. So, as many of us will know, Aedes mosquitoes bite quite often outdoors and quite often during the day as well. And this means they are a particular problem, particularly for people who are going about their everyday lives. So when it comes to thinking about Zika transmission, we do have to think about how we can protect ourselves, how people can protect themselves when they are outside and going about their sort of everyday work and life. So personal protection methods seems to be quite an appealing approach. Can we protect ourselves when we're on the move, essentially? So we've done some work exploring knowledge and perceptions around this, and we wanted to know, basically, first, what do people know about Zika transmission? How do they feel about repellents and other types of vector control? We wanted to model the likely effects of repellents if we were to use them in this sort of scenario. And then part of the work we did as well was to try and develop products which were efficacious, but would also be acceptable to users in this context. So we'll cover the focus group studies first. And we held focus groups with pregnant and non-pregnant women, and in fact their partners as well, in 2 cities in Colombia and in Brazil. And the aim of those focus groups were to explore knowledge and perceived threats from arboviruses, personal and community mosquito prevention methods, and cues to actions, barriers to repellent use, and preferences for new tools. So there are several papers from this work, and I'm going to sort of distill it right down to the main points here. I can't share it all with you. But one thing we found was that several participants recalled no community vector control interventions at all and felt that their community was pretty much abandoned. The accessibility of mosquito repellents seemed to be a greater barrier than the perceived effectiveness or safety. So we always think that people don't tend to like to use them because they think repellents are unsafe. And although that did come up, it was actually accessibility to the repellents that was probably a greater factor in preventing people using them. Interestingly, some pregnant women claimed that the focus group discussion was the first time they'd actually learned about Zika-related congenital abnormalities, which I think is perhaps surprising in some ways, but obviously demonstrates that the messaging perhaps hadn't penetrated down to many people, potentially. Sexual transmission of Zika was poorly understood. Some people felt that the burden of Zika prevention was largely placed on women, and there was quite a lot of misinterpreted information that had been provided, or people distrusted it. So although they saw the information, they didn't really believe what it was saying. Another feeling was that the Zika messaging was too fleeting. It slowed down too quickly and stopped altogether during the epidemic. So we also looked at the Zika messaging itself, and we looked at posters that had been used in the campaign in Brazil. The posters largely focused on mosquito-borne transmission and the need to eliminate breeding sites. So that's good, that's obviously important information, but what we found was that it was lacking in other areas. So for example, talking about the risk of sexual and congenital transmission of Zika and the importance of alternative preventative actions. So from this, we concluded that there is a future need for future campaigns to fully capture the route of transmission and methods to reduce transmission, not just for Zika, but beyond for other diseases as well, potentially. Include tackling the barriers to engaging in preventative behaviors and highlight the benefits of prevention. So although some preventions were sort of displayed on these posters, I think the benefits of using them wasn't perhaps communicated. And also understanding better how to use the technologies that are available. So we have technologies like repellents that are available, but one of the biggest problems that we have is that we don't really know how well those kind of technologies work in these circumstances. And that's a potential area that needs to be improved upon so that messaging can be improved and recommendations can be improved, not just for Zika, but for other diseases going forward. Now, we also explored what sort of product people would feel would be appropriate. And these were the sort of main messages. So people felt that the product should have a non-chemical odor. It should be natural, potentially plant-derived, something that smells nice and would be perceived to be safe. It should be spatially repellent, so be able to protect you. You wouldn't have to sort of cover everywhere, but it would protect anywhere that wasn't covered by it. So normally repellents, you have to cover everywhere, otherwise they don't work. Be applicable to clothing of the wearer's choice. So although we looked at impregnated clothing, people were sort of not really willing to wear clothing that they had to buy that wasn't really their style. So that was really important, otherwise they just wouldn't wear it. And it would have to be low cost and durable with long periods between reapplication. And that's one of the biggest challenges that we have with any kind of repellent at the moment, is keeping it there for long enough. Also, it was felt that educational campaigns should accompany the use of these sorts of tools to avoid misconceptions, particularly around safety. Because although it didn't seem to be the main barrier, it's certainly one of the barriers that should be tackled. Because people believe that repellents are toxic and they're going to make them ill, when in fact there's very much evidence that repellents are safe as long as they're used appropriately. So all of this information has sort of been feeding into the work that we've been doing to look at developing products to protect. And this has been, I'm going to be completely honest, it's been a real challenge. So we've tried to make what's out there better, and it has been very, very hard, and probably because the technology isn't quite there yet. But we have looked at various different ways. So we thought that something that would be good, that would be incorporated into everyday life, would be a wash-in repellent. So imagine washing your clothes with a detergent, and that detergent contains a repellent. Well, that would be a great way to impregnate your clothing and give you protection. Now, the problem was, and we went through a lot of research in different areas here, and the problem was that the repellent washed out too quickly. So the concentration that was left on the clothing just wasn't great enough, and the technology seems to not exist for that. We also looked at treated clothing, so clothing that had been factory treated. And it did work to a certain extent, but again, it washed out far too quickly. And then we had the problems of it not being particularly stylish in some cases, so that wasn't going to work either. So we focused on two other areas. One is a spray-on formulation. So this formulation can be applied to the skin. It doesn't have to achieve complete coverage of the skin because it can work at a spatial distance, but can also be applied to the clothing. So you can just sort of spray it all over. So that is one thing that we're trying and works really, really well. So even just applied to a little bit on the forearm and onto the clothing, you can achieve pretty much 100% protection. We're also working on wearable devices with a company called Vicotech, and these are devices like a band that you would wear on your wrist, again providing a spatial repellence, and we can achieve about, so just by wearing that band, you can protect by about 90%, and using a compound called delta undecalactone, which we chose because it smells nice, it's effective, it's plant-derived, so natural, and sort of fulfills the sort of target product profile that we determined from talking to people about what they might use. So very exciting results there. Some of these things didn't work, some of these things did work, and so we're taking forward the things that did work, and hopefully we'll have some new product types available soon. So another aspect was to look at how well might repellents work in this sort of scenario. So we've done a bit of modeling work to look at different product efficacies, so how effective the product is, different rates of degradation of the product, and coverage levels to determine how many Zika cases could have been avoided if repellents were used. So we've looked at different scenarios here, just as an example. If 80% of the population of pregnant women were using repellents daily, then an estimated 50% of Zika cases could be avoided. So we don't need everybody to use them. And also what it shows is that you don't need necessarily to have 100% efficacy for this to have an impact as well. So that's given us some really good information about how we might exploit this going forward. So in the next part of the presentation, very briefly, I want to tell you about another very exciting piece of work that we've been doing. And this is more around capacity building, and it's called the Global Vector Hub. So this is an online resource. When the Zika outbreak happened, I was very much involved in the response in many ways, advising the UK government, and also other organizations overseas. And I was being asked a lot of questions about the vectors and pathogens of what we know about dengue and various other things in various countries around the world, countries that I've never worked in before. And so I spent a lot of time researching, trying to get the right information very quickly to build up the right sort of picture. And I found it incredibly frustrating, as I'm sure a lot of you probably have, finding the right information, the right data, knowing who to talk to, seemed to be spread across the internet, and took me a very long time to get access to everything. So my thought was, right, we need to fix this and provide something that is a sort of one-stop shop for vector control and vector research. And then I met Trudy Lang from the Global Health Network, and we put our heads together and came up with this concept, the Global Vector Hub. So the Global Vector Hub basically does three things. It provides access to resources, it provides access to data and a repository for data, and it provides access to a network so that you can find out who is who, where in the world, and what is their expertise, linking people together. Just to give you a bit of insight into what the resources section looks like. So it's like going shopping. So if you're looking for a document, it might be a standard operating procedure or a risk assessment or a guideline. You can come here, you can do a search, and just as you would go online to buy some clothes or sort of food or whatever, you can search, you can filter, you can put items into your trolley and then go to the checkout. We don't charge, obviously, it's completely free and open access, but you can then download the documents and you can save them for later. So it's all in one place and very intuitive to use. We've got just short of 300 documents on there already. It's only been launched for a few months, but that is building all the time. And just so you know, one of the most accessed documents was the Global Vector Control Response WHO document, where we've had just over 1,300 views so far. Very excitingly, we've just launched training courses, so 126 training courses in medical entomology from 32 countries in 10 languages, and that's a collaboration with WHO and TDR, and that is now accessible on the Global Vector Hub, which is very exciting. In terms of data, there will be lots of data available that we're pulling in from other data resources and databases. And the idea here is that you can display this on a map, or you can download it and use it for your research purposes. And there will be a function here to help to interpret the data so that anybody, regardless of your level of knowledge, will be able to download a report that will be understandable for you. If you're a policymaker and you want high-level information, or you're a researcher and you want some detailed information. So this is just an example of one of the maps. This is vector abundance, Aedes aegypti in Colombia. And we have other data in there like insecticide resistance, so you can see where data has been collected on that and information on the type of resistance, etc. Pathogen detection, this is the Zika virus and where it has been detected. And even down to bits of information like blood meal hosts, so what kind of mammalian host or other type of hosts are these insects feeding on. So very useful information that can be downloaded there. And we've done various bits of, we've had various bits of impact. So we've run a workshop, again with Global Health Network, that was attended by more than 400 participants from 60 countries to discuss and identify the impact of COVID-19 on the global vector control effort. And really the outcome of that was it has had a big impact and is having a big impact with lots of people being diverted to work on COVID-19. So thinking about vector-borne disease in the context of something like COVID-19 is very important now, but will be important for the future as well. So lots of learnings there that we can take forward. We've also launched a MOOC, so this is a massive open online course on vector-borne diseases generally. So we ran one on Zika already, which had around 20,000 participants. And then we launched another one very recently, more generally on vector-borne diseases, and we've had 4,500 people register for that as well, with really great feedback. So that resource will be on the Vector Hub too. In terms of further outputs, we've developed vector control situation reports for a number of countries. We've done stakeholder analysis on vector control efforts in Colombia, and you can see that we've had a focus on Latin America, but we've extended what we've been doing to other countries as well. We've been able to produce rapid situation reports following natural disasters to share with stakeholders, which have been very well received, for example, after hurricanes and cyclones. We've been, as I say, tackling what we are doing during the pandemic so that vector control efforts are not derailed. So this is the website. I'll put this link in the chat. Please do visit it and have a look around. It is a beta version at the moment. The main version is going to be up very soon, and hopefully that will be a useful resource for lots of us. So I just want to finish by acknowledging all the collaborators, the wonderful collaborators and partners that we've worked with. I can't thank everybody individually, but there have been a lot of people involved in the work that I've distilled into a few minutes presentation there. But you know, what we've learned and developed in terms of personal protection with repellents and capacity building applies to Zika. But as I said, it goes much beyond Zika, and that was always the plan with Zika Plan, was to tackle Zika now, but really to prepare for the next outbreak, which may be Zika, maybe something else, who knows, but there will be something else, and in the context of other disease pandemics as well on a global scale. So I'm going to finish my presentation there. And if I can work out how to do this, I'm going to stop sharing my screen. Has that stopped sharing? I think it has. And I'm going to welcome Kathleen O'Reilly, who is here. Excellent. So Kathleen is a colleague of mine from the London School of Hygiene and Tropical Medicine, and is going to be talking to us about some of the modelling work that she's done, analysing and some reflections on the Zika virus epidemic. So over to you, Kathleen. Thank you.
Thanks, James. Yeah, so I'm going to be talking about some modelling that was largely done in sort of 2017, 2018, looking at the end of the Zika virus epidemic in Latin America. So I'm going to describe very briefly some of the observations throughout the Zika virus epidemic in Latin America. A summary of the previous modelling studies, the rationale that we took in our modelling work and how we use this model. So looking at model predictions. So first of all, what did we observe? And I mean we as a group of individuals who are interested in the Zika virus epidemic. So in 2015, it was essentially sort of the start of the epidemic spreading through Latin America. So for example, the Ministry of Health in Brazil declared a public health emergency in 2015. So this was the start of an increase in the number of cases. And several other countries across Latin America were reporting this new epidemic. 2016 saw a further rise in cases, so almost 500,000 cases were reported across the whole of Latin America. And of course, this was where cases of microcephaly were really starting to emerge quite strongly. WHO declared a public health emergency of international concern, and obviously the Olympics was happening at this point. And then 2017, we started to see a change in the epidemiology. So, well, the latter half of 2016 as well. So there was a reduction in cases. So overall, far less cases were observed. And so one of the questions at the time was, was 2017 just an off year? What's the risk of there being a resurgence in 2018 or additional years? And so this is where mathematical modelling became quite useful. So for those of you who sort of aren't so familiar with modelling as a sort of a research tool, some of the questions that can be answered when modelling epidemics are what will happen next. So here modellers would use the current cases and the model that you would sort of conceptualize sort of mathematically. But essentially what we're trying to do here is that we're trying to capture things that aren't necessarily directly observable. So one of the key sort of examples of this is population susceptibility. So by combining current cases and population susceptibility, you can use mathematical models to develop short and long-term projections of what would happen next. One of the other questions that is quite useful to know is what an impact of a vaccine would have. And this was obviously quite an important question at the time for the epidemic. And of course, you can establish the utility of other interventions. So we've already seen this in the presentation that James just provided. So what are the potential effects of interventions on mosquitoes and people as well? And perhaps a more sort of academic exploration is what drives transmission. So this is quite important if you've got multiple opportunities to intervene in the population, but only limited resources. Which interventions should you target? And you can use mathematical models to help your understanding in what an effective intervention would be. So I think it's fair to say that there were a number of mathematical models developed by a number of individuals across the globe. And so I've just got a very broad summary of some of the main papers on models. And, all of these models had different model structures, different data applied to different populations. But there were three main conclusions from these sort of models. So the first is that there was likely to be exponential growth in the initial epidemic. And then the virus was likely to run out of susceptibles in the human population. And that was one of the reasons why the rate of infection would start to reduce. And so consequently, over the period of about 3 years, the epidemic was likely to be largely over in a majority of locations in Latin America. So what did we do that was, in any way different to the others? How did we add to the evidence base? So what we did, we put together a a mathematical model of the transmission of infection. So everyone in a population would start off susceptible, they would go through their infectious stages, and then they would recover. And because we wanted to model what was happening across the whole of Latin America, we modeled large cities in Latin America, and we included the movement of individuals between cities. We included seasonality in terms of transmission. So this means that we accounted for all of the complexity in mosquito dynamics by having a seasonality curve that was specific for each city and based upon what we know about mosquito reproduction numbers, but also transmissibility and sort of the numbers of mosquitoes that are present. One of the challenges with the data when you're looking at a large population scale such as this is that the data quality was not always as good as we might have wanted it to be. So, for example, we couldn't really compare the rate of infection from one country to another because of issues in reporting rates. So what we did instead of fitting to the time series is that we actually fitted to data summaries and compared this to the the model summaries that came from different sort of parameter values within the model. So it was a slightly different approach to a majority of the others. This slide just illustrates actually each dot is just a representation of each country and also each state in Brazil and Mexico, but actually each large city was modeled in each of these examples. But this figure just sort of roughly illustrates the model fit to the data. So, you know, we use these data summaries so that there was some sort of error in there. But largely this model did a very good job of fitting to data within states in Brazil and Mexico and some countries in sort of the northern part of South America. When we compare the time series across specific countries and specific cities, so here I've just got an example of Aruba, Jamaica, Bahamas, Costa Rica, and 2 cities in Brazil. So you can see you know, in some circumstances, the model fits, which is shown in blue. It represented what was happening in the data really well, which is shown in red. And of course, in other examples, so Aruba being a case in point, we had quite a poor fit of the model in comparison to the data. So this is just sort of an illustration of some of the issues that we encountered. So what did we do with this information? So essentially, what does this mean for 2018 and beyond? And essentially that the model suggested that a vast majority of individuals had already been exposed to Zika virus infection by the end of sort of 2017. And so this meant that in 2018 and beyond, there was a very low likelihood of there being large epidemics, with the couple of exceptions in a few locations. So Argentina, some cities in Brazil, and Colombia were more likely than not to sort of have a reasonable number of Zika virus cases. And beyond 2018, the infection rates were predicted to reduce further. So in conclusion, it's a very broad overview here, but the highest number of cases were reported in 2016 especially, and as we've seen with other aspects of today, the focus should be on treating individuals that were affected at this time. You know, this does present an issue for things such as vaccine challenge studies, and it also illustrated some challenges with surveillance. So when you've got disease surveillance at this sort of continental scale, there's lots of data, but this, you know, it doesn't necessarily mean that you've got lots of information. And so, you know, this is a really good sort of example of where in some cases we do have a long way to go in ensuring that we have accurate and informative disease surveillance for infectious diseases. And yeah, just a final note that this work is summarized in a paper and the link is just there at the bottom. So yeah, thanks for listening, and that's the end of me. Thanks.
Not the end of you, I hope, Kathleen, but the end of your talk, perhaps. Okay, so do we have any questions for myself or for Kath? I can see in the Q&A that it is blank. So it's either stopped working or everyone's needing a break or doesn't have any questions, which is absolutely fine. Maybe I'll ask a question to Kathleen. I just wondered, it's quite an open sort of question, but what would have happened if Zika happened now? So during a pandemic like COVID and the Zika.
Yeah, I think it would be very challenging. Like, the first thing is, how quickly would we detect it? So it's very apparent that in other infectious diseases, disease surveillance has really reduced. I spend a lot of time working on polio. Disease surveillance for polio has massively reduced across the entire world because, you know, a lot of health professionals are quite rightly focusing on COVID. So you know, there's this difficult balance. I think the other thing is the change in how people move and sort of their behaviours and what impact that is likely to have on vector-borne diseases. For something like Zika, it's a bit difficult to tell. You know, it really depends upon the location. You know, a lot of people are staying at home a lot more. So then it depends a lot more on what their interventions are in the home. And that's going to really vary across the board. I mean, it's a good reason to sort of carry on doing good disease surveillance and understanding how pathogens transmit.
Yeah, absolutely. And I guess the big challenge, which is what we've seen, is whether that surveillance continues, whether there are enough resources to continue that sort of surveillance through a situation like this, a pandemic.
Yeah.
Okay, so we've got a couple of questions now. Do your models also predict an outbreak-free time after an epidemic? That's a question from Charlotte.
So, I mean, a lot of, well, the model sort of suggested that a lot of people had already been exposed to Zika. So, you know, with making some very simple assumptions, and I'm sure there's plenty here that might want those assumptions to be quite different. If you assume that, you know, it's previous infection causes long-lasting immunity against Zika virus and future Zika virus infections, it's unlikely that Zika is going to re-emerge in the next 20 years or so. But like I say, you know, if that's only if your current immunity causes lifelong immunity. So, for example, if the Zika virus was to change substantially so that your current immunity is less effective, then that could change things. And also, you know, if there's populations that weren't infected, they might be quite small in number, so it's less likely. I think, you know, most seroprevalence studies illustrated some quite strong responses against Zika virus. So it does suggest that future epidemics, you know, in the next 20 years are low. I wouldn't say that there's no chance, but low.
And do you mean that from Latin American point of view, or do you mean that sort of globally? With that?
Yeah, I think, you know, I can only really make comments on Latin America, really. I think we've seen from other researchers today that you know, the biology of the Aedes aegypti, but also the virus in general, is quite different, as, you know, Africa, Asia being cases in point.
Yeah, absolutely. Okay, so we've got another question here. Is passive surveillance enough for the detection of a highly asymptomatic Zika infection?
Yeah, it's a very good question. I guess, yeah, how best to answer that one? I mean, you know, it should be a bare minimum. I think there are things within passive surveillance that you could do a better job of. So, for example, looking at serological responses in blood donors, I think has been extremely useful for COVID. That's illustrated its use. There was a really interesting study in the UK, I think published today, and I think that sort of passive surveillance does still have a very good use. It does, you know, there's always a delay, so I think some form of active surveillance is worthwhile, but, you know, there's a resource question, and that does need to be balanced against, all the other pathogens that are out there, and the pathogens that are causing a public health problem. And of course, things such as dengue are also a big issue. Combining surveillance for multiple diseases obviously makes a lot of sense, and I think that would be a very useful approach.
Yeah, it'd be useful to know what's actually being done now from that point of view, actually. So there's another question from Anonymous, although I think it's Annalise. And it's, maybe we've addressed this already, but I don't know if you've got anything to add to it. But when do you think Zika will come back again? When do you think it will come back again?
Yeah, I guess, as a major epidemic disease where, there needs to be sort of multiple country interventions, potentially not for a long time. I think, it's, whilst modelling is sometimes referred to as a crystal ball, actually, those kind of questions are very difficult to answer with models because it isn't a crystal ball. And, that shouldn't mean that things such as vaccine development shouldn't be, stepped up and have the potential to be made readily available should something like Zika happen again.
That's great. Okay, so I don't think there's any other questions, and we're pretty much bang on time. So I'm going to close this last session and thank Kathleen very much for a great presentation, and thank you to the organisers as well. Thank you.
Well, thank you, James and Kathleen for excellent presentations. I think we have had 3 very interesting sessions as testified by lively questions and discussions. Zika Plan has addressed many questions, but you all agree that many questions still remain unanswered. And I think the question when it would come back is an important one, and therefore, Kathleen, your modeling is important because it does drive vaccine development and financing of vaccine development. So building sustainable networks and the capacity to respond to the re-emergence of Zika in the near future or in 20 years' time, or the emergence of any other disease is really, it was and is the core of Zika Plan. But this can only be done in collaboration with other research networks, in particular with some of the other Zika consortia. And let me acknowledge here the tremendous collaboration we've had over the past years with Zika Action and Zika Alliance. Only together with them, we can build a legacy that will last beyond this funding period. May I draw your attention to Zika Plan's website? Just Google Zika Plan and you will find it. Please have a look at all our activities, at the experts, at the sites that we have, the networks, but also click on publications so you can see and evaluate our scientific output through these scientific publications. In this context of the website, please allow me to thank Margot Luciano and her team from the Fondation Mérieux for creating and maintaining our exciting website, and also for all her efforts in writing our newsletters. And currently we're working on a series also of videos where she is responsible for. Allow me also to thank Coran Woolman-Tardy, who served as our Zika Plan communication director since Zika Plan's inception, until she was invited to become the head of the US branch of the Fondation Mérieux. So thank you all. Now a few announcements. So today was our first of 3 public webinars. Our second Zika Plan dissemination event will take place on 10 March, again on a Wednesday at the same time. The title for the next one is Zika and the Nervous System, Emerging Lessons for Emerging Viruses. It will include a series of short new films with a focus on EuroZika. And as you know, Zika not only affected pregnant women and their offspring, but also adults in the sense of unusual neurological complications. And then the third and our final webinar will be on 24 March, also on a Wednesday afternoon. And during our final webinar, we will showcase the networks that were formed because of Zika Plan's funding. And these networks pertain to diagnostic platforms, the neurological networks, the birth defect surveillance, networks for training and capacity building. And remember, after all, our motto for Zika Plan was and is from urgent response to long-term capacity. So we will run that final seminar together with Zika Action and Zika Alliance. Now, let me also express my gratitude to the European Commission for funding us and for their ongoing support throughout the project. It was very time intensive, and we delivered. Let me thank also the Global Health Network for putting this event together today, especially Bonnie Baker. But I also would like to thank the Umeå University for hosting the consortium and for their financial oversight and project management. There's so much work in the background. Here, I would particularly like to highlight the tireless efforts undertaken by Dr. Raman Prit as our Zika Plan Executive Director. It is really because of her that Zika Plan achieved what it has achieved today. So we finished punctually. We did our job for today. I hope you enjoyed it. See you again next time for the second on the 10th of March with the entire Neuro Zika team for a Neuro Zika event. Goodbye and thank you to all.